Hello, good afternoon to all, or good morning or good evening, depending on where you are. I would like to welcome you all to the workshop on FEEDNETICS for aquaculture researchers. We organized this workshop at Sparrows so that you can have a demonstration to see how this type of nutritional tools can assist in the study of aquaculture nutrition. First of all, I would like to thank you for your interest and for joining us in this workshop. We hope at the end of these 2 hours you'll find it an interesting couple of hours. Just a bit as an introduction, just to let you know that FEEDNETICS is part of the Sparrows development effort of what we call the nutritional tools. So basically, information technology tools which can be used to help studying feeding and nutrition, and also to optimizing it at the farm level. This has been one of the lines of R&D investment of Sparrows in the last at least 5 years. At the moment, we have 3 persons working full-time on this, plus 2 PhD students, and some others that also collaborate, like myself, from time to time. So we hope that this will be an interesting afternoon, as I said, and I will then pass the word to Tomé Silva, who will give you an overview, and then after that we'll have 2 use cases. So we thought that the best is to show you some examples on how FEEDNETICS can be used, first to evaluate amino acid requirements, and second to compare novel diet formulations and predict long-term effects under more farm-scale conditions. So let's say to do an extrapolation from a trial of a few months to up to a full production cycle. We would be happy if you have questions to make, especially on these on FEEDNETICS in general, and on the use cases in particular. We'll have 2 Q&A slots in the program after the 2 use cases. But we'll ask you to write the questions in the chat on or the Q&A option, even better, of Zoom, and then we'll come back to these questions during this Q&A sessions. Okay. So without further delay, I will pass the word to Tomé Silva, who will give you the FEEDNETICS overview, and then we'll continue with the use cases. Tomé, please.
Thanks, Luis, for the introduction. So as Luis said, I will start with an overview of FEEDNETICS. And so FEEDNETICS is a virtual environment for aquaculture, and it allows you to, in general, to evaluate the effect of aquafeeds, farming conditions, on different aspects of fish, different indicators of fish biology, such as growth, feed conversion, feeding costs, economic conversion, body composition, and waste emission. It has a wide range of practical uses. For farmers, for example, it's a tool that can be used to optimize feeding, both in terms of which diets to use and the feeding rates. For feed producers, it's also a tool that can be used to assist in customer support. And in the academic context, it can be used for different things. For example, if you're planning a trial, a fish nutrition trial, it can help you determine an optimal design for your experiment, and also to assist you in the interpretation of the results of your trial at the end. Furthermore, it can be used to interpolate or to extrapolate trial results. So you can try to estimate what would happen in terms of long-term effects based on the short-term experiment. And in general terms, it's a useful tool that can be used in teaching activities. In a global sense, FEEDNETICS as a tool is based on a mechanistic fish growth model that's mostly driven by temperature and feed, both in terms of quantity and qualitative properties of the feed. And it simulates over time fish growth and composition, and based on that, a series of relevant indicators. One aspect that sets FEEDNETICS apart from similar options is the fact that it takes into account the effects of amino acids in all these in growth, composition, and all these indicators. If you look more specifically into the model that drives FEEDNETICS, it's a nutrient-based model, and like all nutrient-based model, growth is modeled as as the result of or growth is driven by the nutrients that we provide in the diet, and results from the partition of these nutrients into a retained fraction and a lost fraction. And this happens under the control of metabolism that enables the conversion between the different nutrients. Of course, because this is a mechanistic model, we try to ensure that it replicates accurately what we know about metabolism, about fish biology, and about physical processes in general. So aspects such as, for example, mass conservation law and energy conservation law, these physical aspects are very important when you're developing a mechanistic model. In general, a FEEDNETICS model, or technically FEEDNETICS model, is a system of differential equations. But if we look at it from a high-level view, we can more or less separate it into a farm model, what we call FEEDNETICS farm model, and within this model we have the FEEDNETICS fish model. This fish model is driven, as I said, by temperature and aspects that have to do with, on one hand, the quantity of feed, and on the other hand, properties of the feed. So its content in terms of macronutrients, micronutrients, and also digestibility coefficients, and so on. And then the fish model, which is a multi-compartment model, is basically being driven by these inputs and continuously updates its state variables, body weight and composition, based on these inputs. And based on these state variables, it provides a series of indicators. Most of the, let's say, interesting part of this model happens in the body compartment, where the metabolic processes are modeled, let's say. If we look at these metabolic processes a bit in more detail, FEEDNETICS tries to model the most important aspects of metabolism, of central metabolism, and it explicitly models amino acid and fatty acid metabolism. So on one hand, we have a quite detailed amino acid submodel, where we consider, for example, amino acid interconversion reactions. And then on the side of the fatty acids, we consider a more simple but equally effective dilution model. And so it's precisely the fact that it has this detailed amino acid submodel that it's able to take into account the effects of amino acids on fish growth and so on. Of course, we have this general model structure, but then to make the model work for different species, we have to calibrate specific parameters of the model for the different species. And this takes some effort and requires lots of data. And so for now, the species that we have available and that you can use FEEDNETICS with are the seabream, seabass, trout, and tilapia. And besides that, another option and something that we work on as well is if you want a specific calibration for a species that we do not have available, or a specific calibration that's suited for your specific production system, imagine, that's also something that we can do and that we have done before, as long as there's data available. And then another, let's say, important aspect to consider is that a model is only as useful as it's capable of predicting things. And so after we calibrate the FEEDNETICS for each species, there's an important step of validation that we undertake where we compare model predictions against, for example, we run the model with inputs from trials, for example, from the literature or from other trials that we have, and we compare the predictions with the measured outcomes. And so just to show you some examples of some comparisons that we've done in that sense, to show you how we can, for example, use FEEDNETICS to predict the outcome of nutritional trials. So a first simple example is that, for example, we can take a trial from the literature where a simple trial where a single factor, nutritional factor, was varied, and we can try to see if FEEDNETICS is able to accurately reproduce the results of this trial. And we can see that quantitatively it's quite close to what was measured, but most important than that, qualitatively the model predicts accurately the different trends, what happens when we increase fat in qualitative terms. Another example, this time from a project in which Sparos participated. Here we were trying to estimate the requirements for methionine, basically, and so we fed fish with increasing levels of methionine. And what you can see here in this example is that if you try to replicate the growth curve of this trial for the different tanks using a simple nutrient-based model that does not account for the amino acid composition of the diet, you get something reasonable but not perfect, let's say. While if you use a model that does account for the effect of amino acids on growth, you get a much more accurate prediction. Here is a more complex example, because here 6 different sources of protein were tested. So these sources were not just changing one factor, we're changing not only the amount of protein, but also the proportion of different amino acids in the diet. And we see that even in this challenging context, FEEDNETICS manages to predict quite well what happened. And then just to finalize, to show you what happens when we try to predict longer term trials, which is a challenge because in this context, errors tend to accumulate. And what we see is that in this case, for example, comparing against trials reported in the literature for seabream and seabass, we see that, and it's important to notice that these are very long-term trials, more than 1 year. And we see that although, of course, predictions are never perfect and data is never perfect, we still get a very good long-term prediction of growth. And then just a final example, additional example, in this case, within the context of a Valormar project, within the context of this project, we were trying to evaluate whether FEEDNETICS is able to predict long-term growth, but in an actual commercial farm, in a commercial setting. And we were able to demonstrate that indeed the results are quite good, even over the course of a very long growth trial. And so after this quick overview of FEEDNETICS and the FEEDNETICS tool, we now will talk a bit about two more concrete use cases for FEEDNETICS. And the first one is to talk about how nutritional models such as FEEDNETICS can be used to evaluate and help estimate amino acid requirements. And so I will start first by giving some context about the problem of the estimation of amino acid requirements. In this context, it's always good to start with a definition. And so when we talk about nutritional requirements, particularly in animal production, the working definition is usually something like the amount of a nutrient required for an organism to express its full growth potential while ensuring good health, welfare and health. So there's an emphasis on the productivity side. And in practice, if we want to determine the nutritional requirement of an essential nutrient, there's many ways to do it. And the first step is usually to just go to the scientific literature and read what has been already described about this subject. But of course, this information has to come from somewhere, and most of the information comes from experimental approaches, from measuring things under very controlled settings, what's known as dose response studies. But there are also other ways of estimating nutritional requirements. For example, using observational approaches where you have a less controlled setting. So for example, when you do a meta-analysis, or when people use self-feeders to try to determine nutritional requirements. Furthermore, as you'll see later, there are also modeling approaches. You can also use nutrient-based models to give you some help here. Either way, because experimental approaches are the most important and in some sense the gold standard of determining nutritional requirements, my presentation focuses on those. And when we talk about experimental approaches, almost certainly we're talking about performing dose response studies. And here what we do is, if we want to determine the nutritional requirement of, let's say, an amino acid, we would provide fish with different doses of this amino acid and measure how a certain relevant response is affected by these different doses. And when I say relevant response, what I mean is that it should reflect what's written here. In some sense, it should reflect growth and also welfare and health. And in practice, in aquaculture, this response is usually nutrient retention or growth, not only because they're directly relevant, but also because they're generally an indirect indicator also of welfare and health in some sense. And so looking at the results of some of those response studies, what we see is that at the end of a dose response study, what we obtain is something like this, where we have the result of a certain response measured at different doses. And this allows us to recover the dose response curve or the dose response relationship. And when everything goes okay, this allows you to answer your question. So in this particular case, we can see that looking at the dose response relationship, we can say that we need to include at least 42% of protein in the diet of these fish to ensure maximum growth. So this would be the nutritional requirement for protein. One thing to have into account and that we should pay attention to is that we can only obtain reliable estimates if these doses that we measure are well distributed. And we see that in this case, that's the case. We see that we have a high number of doses that were tested, and they're balanced in the sense that 3 doses were tested above the requirement and 5 doses were tested above the requirement. So, and at least 1 dose was measured close to the requirement. So we can say that in this case, we have a very good result precisely because the doses that were tested were well chosen and well distributed. To give you a slightly different example, in this case, we're determining the nutritional requirement of phenylalanine, which is an amino acid. And what we see in this case is that there's some level of ambiguity, meaning if we choose a specific dose response model, we get a certain estimate. But if we choose a slightly different model, we get a different estimate. And so it is not exactly clear what we should consider as the requirement. And what we see in this case is that we get these unclear estimates precisely because we do not have a very good distribution of our doses. You'll notice that most of our doses are basically below the requirement, and there's only one dose over here measured above the requirement. And so because there's not a good distribution, we do not get very consistent results. And so when we talk about the challenges of those response studies, there are many. One of them is the one I just showed, which is you need a good trial design. We need to test several doses of the nutrient you're interested in, and these have to be well distributed. And this is very important to have good results. And so in practice, if you want to study the requirement of a certain nutrient, you need to have some sort of, need to have a good guess or at least a rough estimate of what that requirement should be, which is slightly annoying, but it's a fact. Another big challenge is the fact that nutritional requirements depend on context, meaning we may have determined what's the nutritional requirement under a certain body weight and under a certain temperature and under a certain rearing system and so on. But this might not be universal. If we change the context, the nutritional requirements can change slightly. So the challenge is that to have a good characterization of nutritional requirements, you need to do more than a single trial. You need to do several trials under different conditions. So this is a big challenge in this context. And finally, another big challenge is that the requirements of nutrients often depend on the doses of other nutrients, meaning on the presence of other nutrients in the diet. And what people usually say to describe this effect is to say that nutrients interact. And in practice, when we're studying the requirements of essential amino acids, this is a big problem. So to see why, we should look into detail at what it means for nutrients to interact, talk about nutrient interactions. And a good example of that is what happens with lysine and histidine, or almost any other two essential amino acids. And in this case, what happens is that these two nutrients are both required for growth, and they're not interchangeable. You cannot, most organisms cannot convert one in the other. And so the ratio at which you provide them in the diet is very important. To see this, we have to consider what can be called the multivariate dose-response relationship. So here we're not just changing the dose of a single nutrient, but we're changing the dose of two different nutrients and seeing how a response is affected by that. And to do so, because now we don't have a dose-response curve, but a dose-response surface, we need an additional dimension. And you could represent it with something like this, where you can see that if you provide low amounts of histidine and lysine, the growth is low, the response is low, and if you provide large amounts, the response is high. A more convenient way of displaying the same information is to just see this surface, to look at this surface from above, which gives you something like this. And this is more convenient because you can focus on the doses, and the response is simply the color that you see. So in this case, we can see that the plateau, the maximum growth that we see here corresponds to this dark red area over here. What's particular about this type of interaction is that, again, the ratio between the doses is important. And so when we consider what's the minimum amount of histidine and lysine that we need for maximum growth, we see that we always have to travel along this line over here, which corresponds to this ridge that we see over here, which corresponds to the slope of this line corresponds to the optimal histidine and lysine ratio. Now, it's important to consider what's the consequence of this interaction existing between lysine and histidine. So imagine we want to measure lysine requirements. So we're going to do what we said before. We're going to measure the amount of what happens to growth as we vary the amount of lysine as we travel along this line. If we choose to do it over here, so meaning we're choosing to do it with diets with low amounts of histidine, what happens is that quickly we hit the growth plateau over here, and it seems like the apparent requirement for lysine is very low. If you increase past here, you do not get more growth. If you try to do exactly the same thing, but using diets with a higher amount of histidine, so you test different levels of lysine, but with diets containing medium amounts of histidine, you'll see that the apparent requirement is higher than what you estimated before. And if you repeated this with even higher levels of histidine, you would estimate an even higher apparent requirement for lysine. And so what we see, and the big problem here, is that the requirement estimates that we're getting for one essential amino acid, in this case lysine, depends on the amounts of other essential amino acids, in this case histidine. And this is a problem, obviously. The way that people usually try to solve this problem, the most direct solution would be to try to study this whole surface. So you would do a full factorial design, multivariate design, for example. But in practice, this is something that's very expensive. So as we've seen before, if you're studying a single nutrient, you need to have at least, let's say, test at least 5 or 6 different doses of that nutrient. And if you're not doing any replication, that implies at least you're going to need 5 or 6 samples, meaning 5 or 6 tanks. If you're studying 2 nutrients, like we've just seen in this histidine lysine example, if you're going for a full factorial design, meaning a 5x5 or a 6x6, without any replicates, you're already going to need 25 or 36 tanks. And if you go to 3 nutrients, then it becomes, the numbers quickly become absurd. So in practice, this is not the solution that people prefer to use. The solution that people prefer to use is to exploit the structure of the nutrient interaction. And by this, I mean, if we go back and look at, since we know how the interaction works, we can take advantage of that. And if we look back at that interaction that I've shown between histidine and lysine, and focus just on the part with high levels of histidine, we see that as long as we're in this part of the dose response surface, and we try to measure the requirement of lysine, we always get the same estimate. So as long as the level of histidine is sufficiently high, the specific amount doesn't really affect our requirement of histidine. And the same happens if we do the opposite. If we try to estimate the requirement for histidine, as long as the levels of lysine are sufficiently high, it doesn't really matter what's the specific level of lysine. So in practice, the solution that people use is, if I'm studying a certain nutrient, I have to ensure that there's an excess of everything else that I'm not studying. In practice, this is the solution that people use to deal with the nutrient interactions when studying amino acid requirements. And so, in summary, when we want to experimentally determine the requirement of one amino acid, we use those response studies. But as we've seen, we need to start with some sort of guess or rough estimate of the requirement of that amino acid just to design the experiment, because we need to know more or less where the requirement is so that we can put sufficient amount of samples below and above that requirement. And as we just seen right now with the issue of interactions, because these interactions exist between nutrients, you also need to have, even before you start an experiment, to have a rough idea of what are the requirements of other essential nutrients you are not studying, just to ensure that these nutrients are not being limiting factor and affecting your estimations of the nutritional requirements. And so to wrap up and conclude this presentation, of course, experimental approaches are the gold standard of the study of nutritional requirements. But as we've seen, when you're designing those response studies, you need to already know something or guess, if you don't know, something about both the requirement of the thing you are studying, but also the requirement of things you are not studying due to the existence of these nutrient interactions. And so, although experimental approaches are, let's say, the gold standard, having all sorts of prior information that we can get from complementary approaches are extremely useful, not only when designing the dose response studies, but also when interpreting the dose response studies. And so, having said this, and having talked about experimental approaches, the next presentation is specifically to illustrate how the modeling approaches can be useful in this sense and give you complementary information to undertake better trials and to better understand the results of this trial, of your trials, when you're studying nutritional requirements. And so that's what Anna will now speak about, give you a concrete example of how you can use nutritional models such as FEEDNETICS in this sense. And in the meantime, if you have any question, please use the Q&A option, and then we will get back to you during the Q&A session. So please, Anna.
Thank you very much, Tomé. Hello to everybody, and thank you for coming. And Tomé, thank you very much for this, for your clear overview about the definition of the amino acids requirements. During the next few minutes, I will present a use case to illustrate how FEEDNETICS can be applied to leverage your research about this topic. During the presentation, please use the Q&A button as mentioned by Tomé, and let us know specific comments and questions that will be addressed and taken during the discussion session, which will be moderated by Jorge Dias. So within this presentation, we will focus on illustrating how FEEDNETICS can support the fine-tuning of the trial design and the interpretation of the trial results, looking into a specific use case of a dose response trial for methionine requirements definition. And for that, our FEEDNETICS application will consist on simulating a dose response trial for methionine for the seabass based on a study by Tully et al. That examined the fish performance regarding weight and end gain fed different dietary levels of meth. Next, and also very important step in a modeling application before going into the analysis and results interpretation is to evaluate how the model outputs are comparing with the data. And finally, we will see how we can use the model results to interpret the trial outcomes, and also how they could be used in a pre-trial stage to fine-tune the trial design. And just before we move to the next slides, we would like to thank to Francesca Tully for all the support that allowed us to run this simulation with the detailed data from her trial. So for the simulation, we have considered the trial conditions for the several treatments as per the published data. This slide shows the consolidated trial data that we need to insert in FEEDNETICS. And next, we will show how how you do it. And this is important to give a practical idea to those that never used FEEDNETICS, that running a model like this is accessible to any aquaculture researcher with no need to be a modeling expert. We open the web app and come to the input data and farming conditions section and create a new farming condition for each treatment. And you see here a few of them already created in these lines. Let's choose this diet 2 for diet 2 and tank 16 to see what we need to set up. Let's start with the initial conditions, where we insert the 50 fish of 13.7 grams, which were the specific conditions for this tank. Then we go to the population section to enter the number of fish recorded over the experiment. And at this population menu, you need to download a spreadsheet file by pressing here the export button, fill in the number of fish along the period of the trial in the spreadsheet file, and then import it back to FEEDNETICS to load the number of fish. After doing this for all the tanks in the trial, we are done with the farming conditions for this use case and can proceed to insert the temperature. Like for the farming conditions, you create a new temperature and insert your data by exporting and importing back a spreadsheet file. And after that, after inserting the trial conditions and temperature profile, we can move to the feeds and feeding regime. And because FEEDNETICS is a very detailed nutrient-based model, as explained by Tomé, the feed data to insert includes also, among a wide range of properties, includes also the amino acid profile. And so for simulating the MET-U9 dose response trial, we had assumed the exact same values for proximal composition and the MET-U9 levels that were obtained in each diet by Tully et al. And for the remaining parameters that are not available in the paper, such as digestibility coefficients, we have estimated them from the ingredient composition or assumed a default. For the feed ration, we have considered the daily feed quantities that were given at each tank. So to insert the feed property data in FEEDNETICS, we go to the input data and feed sections and create a new feed for each diet of the trial. Here in these lines are already inserted all the diets that were used in this specific trial. For each diet, we need, let's choose here this one, we need to fill in each of these menus. These are proximal composition, digestibility, and the amino acid profile. And as you can see with these print screens here, you will be asked to insert the parameters that you typically analyze at your experiments, or that might be estimated based on your feed formulation. And finally, after having filled in the diet properties, we need to specify the feeding regime for each treatment and tank. Here, at the explicit feeding option, you introduce the daily feed quantities that are registered during the trial, using also a spreadsheet template that you can export and import back with your data. And after you have done all this data insertion, input data insertion, you finally can move to prepare and run the simulation. Let's look for one of the trial tanks and see in what consists these simulation settings. So it is here at the simulation settings where you select the species that you are using, and combine the data that you have inserted previously for the trial conditions, the feeding regime, and the water temperature. And finally, you specify the start and end dates of the trial. And after we run all the simulations for the dose response trial, which in this case correspond to 24 simulations, we can finally go to the results section. So let's choose the control tanks and see if the fish grow as expected by selecting the stock cluster of indicators. And here I've chosen the average body weight indicator, and we can see here the growth curve for the 3 control tanks that we have just selected. And looking into this chart right after running the simulations, it is very important because you can check if everything is running okay. Sometimes there are mistakes that are made entering the data, and this way you can spot immediately and fix whatever is needed before moving to the results analysis. In this case, it seems like everything is looking good. These points here, highlighted here, of decreasing growth correspond mostly to the sampling days where no feed was given, so it matches what is expected. And if we reach the end of the simulation with a final fish weight of around 45 grams, and just to have at this stage, just to have an overall view, let's look into the average final weight reported by Tully et al. For the control. It is around 44 grams. So it looks like the simulations ran as expected, and we can now focus into the detailed model evaluation. And the best way to do the analysis is to export the outputs into a spreadsheet file. First, you select the simulations up to 5, and then export by pressing the book of reports. You need to do this for all the tanks of your trial, and you will get an Excel file that contains all your outputs. In this case, we want to further analyze the data to consolidate the results for the 24 tanks. So we are interested in this sheet here, the general output, with all the raw outputs of the simulation. At the next use case, Philippe will show you other sheet of this file, which presents the results in a more user-friendly way. An important note at this stage is that FEEDNETICS produces many outputs per simulation, many indicators, providing you with a huge potential to carry out many types of analysis. So it is very important that you keep a focus on what are your research objectives. And in this case, our first one is to compare the model results with the data concerning first the average body weight. And so with the model results and the data, we built this plot of observed fish body weight versus the simulated fish body weight. And we also have here the calculated error metrics. And we can see here that the control diet, which are these black dots here, the control diet exhibits the lowest MAPE, showing a good fit between the model and the data. And for the treatments with the semi-purified diets, larger deviations were obtained, in particular for these diet 2 and diet 3. In this particular case, a possible reason might be the difference in the efficiency of utilization of the crystalline amino acid by the fish when compared with the efficiency of utilization of the amino acids from intact protein. So next, because we wanted also to compare the MET requirements obtained from the trial results with the requirements from the model results, we compiled these charts of those response using the broken line approach as done by Tully et al. To estimate the requirements, in this case also by the model outputs. And here we observe a difference between the data and the model results, which is the consequence of the differences in the estimation of the body weight for the semi-purified diets. And when interpreting the comparison between the model results with the data, we should always remember that what the model gives us is what is expected in theory, which for a number of reasons might differ from reality. And when we have a difference, that might lead us to raise more questions to be further investigated, which in this particular case might be related with the impact of the amino acid source in the definition of the methionine requirements. So here we have the two charts for the dose response trial with both the trial results and the modeled results. And we can see that the pattern of the dose response trial seems to be well simulated, which is an important and an indication that we can use FEEDNETICS to provide the prior information that Tomé mentioned for the optimal design of a dose response trial. Let's see how in the next slides. So this table presents the model results at the end of the trial, and it indicates that the diets with the MET levels of 1.6 and 4.9 gram per kilo of the diet exhibit the worst fish performance as expected, while the treatments with the MET levels equal or greater to 7.3 gram per kilo of the diet showed similar fish performance. If we had carried out a similar simulation like this before the trials, we could use the model outputs for its planning in the following ways. For instance, to reduce the number of treatments and thus the number of tanks by removing, in this case, for instance, these two treatments with the higher MET levels. Or for instance, if the number of treatments or tanks is not an issue, we could redesign the trials to test lower dietary MET levels, considering these intermediate levels around the simulated requirements, which will fine-tune the dose response curve that will be obtained. And so to wrap up this use case, the key take-homes from this application are that at the planning stage, FEEDNETICS can support the trial design and the definition of the dietary treatments, providing that prior information that's important to set up a dose response trial. And after the trial is carried out, FEEDNETICS can help to interpret the results by allowing the comparison of what is expected in theory with what was obtained in reality. And further, and also very important, is that by analyzing the model results, you can raise new questions to be further tested in further trials or further investigated. And actually, this use case is just the tip of the iceberg concerning the dietary methionine and total sulfur amino acid requirement definition. As further discussed by Tully et al., the requirements that they determined at the paper are different from other publications where other cysteine and protein levels of the diets were considered. And so a further FEEDNETICS application could explore those effects on the MET or TSAA requirements. And the MET and the cysteine interactions show this particular behavior compared with other essential amino acids, whereby they are interchangeable. And with a model such as FEEDNETICS, it is accessible, though laborious, but accessible to explore a wider range of this MET and cysteine interaction surface. And this is something that for which we invite the students that are listening to us and that work on this subject to apply to the FEEDNETICS student prize. Follow this QR code to see the regulations where they are entitled to use FEEDNETICS for a topic of their choice subject to a first ranking, of course. And thank you very much for listening.